Now I'm going to talk a bit about some of these organs, some of these glands that only serve the endocrine system and aren't serving another function. We're going to concentrate on those for the remainder of this endocrine system, and then go ahead and the rest of them are still important, but we're going to be talking more about those as they interface with other physiological systems. So we're going to come back to those, but right now I need to take the time to really concentrate in on these ones. And the first one I'm going to talk about is the pituitary gland that really is, in a lot of ways, very intimately connected to the hypothalamus. So I'm going to talk to them together. The pituitary gland here is a lobe essentially off the brain here, sitting right here. Then the hypothalamus is actually this area that's sitting kind of right on top of that, right up here. So they're kind of sitting pretty intimately together right there. So let's talk a little bit about the general anatomy of the pituitary gland. And one thing just to know, because this will come up, is it's also called the hypophysis. So it kind of sounds like you're trying to say hypothesis with a slur. So it's also called the hypophysis, and it's going to be connected to the hypothalamus by this branch, this small limb called the infundibulum. Now, the hypothalamus is going to control most of the pituitary release of hormones. So the hypothalamus is really in control of much of what the pituitary is doing. And the pituitary gland is really broken up into two separate portions that are really both anatomically and functionally very different. The first one is what's known as the anterior pituitary, which is also known as the adenohypophysis, which is connected to the hypothalamus vascularly. So it actually has blood vessels that are going between the hypothalamus and the anterior pituitary that serves kind of as a communication route between those two. Also, there's a posterior pituitary, which is also known as the neurohypophysis. And this is connected to the hypothalamus neurally. So there's actually neurons that are traveling between the hypothalamus and the posterior pituitary. Let's give you a little, another little glance at how this is happening. So as you look at this, again, this blue in here is the hypothalamus in this area. It's connected to the pituitary by the infundibulum, this area here in orange, and then down below we have the two branches of the pituitary, the anterior and the posterior. So that's that. So let's talk about each of these lobes in succession here. So first let's talk about the posterior pituitary. And together with the hypothalamus, this really forms a particular neuroendocrine system. This is one specific neuroendocrine system where we have the nervous system and the endocrine system that are married in very, very intimate contact. And the reason this is because we have axons from the hypothalamus that are going to terminate on capillaries inside the posterior pituitary. So we have these neurons that their cell bodies are in the hypothalamus. These axons are extending down to the posterior pituitary, and their axons are terminating on these capillaries where they're going to dump their neurohormones into this capillary. So really, the posterior pituitary is really just an extension of the hypothalamus. It's essentially this lobe of this hypothalamus that's extending down to the pituitary gland so it can dump some of its neurohormones into the blood supply there. In particular, the hormones that this is going to be doing secreting is vasopressin and oxytocin. They're going to be stored independently in these secretory granules, which are essentially secretion vesicles, in the axon terminals, and it's just going to wait there until the release. Let's look at kind of a graph of how this works. Up here we have the hypothalamus, and like I was saying, up here we have these cell bodies of these neurons. Their axons are going to be extending down to the posterior pituitary here, where they are synapsing onto these capillaries. They're going to release neurohormone into these capillaries, the vasopressin, the oxytocin, that will then be carried away into the bloodstream. So that is specifically what's happening with the posterior pituitary. Let's talk a little bit about these two different hormones that it is secreting. The first being vasopressin, and vasopressin is also known as antidiuretic hormone because it blunts the urination of animals that it's active in. And it's going to do this by enhancing retention of water by the kidneys. And under normal conditions, this is the hormone that is actually regulating water balance in the body. So both water balance being excreted in the urine, how much is being exported from the body that way, but through that is going to maintain the water balance of the body itself. So this is really important for that. It's also going to cause contraction of arterial smooth muscle. So certain types of arterial smooth muscle are going to contract, and this is going to cause an increase in blood pressure, therefore the vasopressin name. Okay, oxytocin, we're not going to talk a lot about right now, but oxytocin is involved in various social bonding actions, and then it also stimulates contraction of uterine smooth muscle during parturition in this positive feedback loop. So as you get uterine contractions, this is going to stimulate the release of more oxytocin, which in turn stimulates more uterine contractions, and this is going to actually this positive feedback loop between oxytocin and uterine contractions is going to be what eventually causes labor to begin and causes the delivery of the child. Okay, so let's move on to the anterior pituitary here. And the anterior pituitary is really a very different animal from the posterior pituitary. It's connected to the hypothalamus through a vascular portal system. Now, a vascular portal system, there are several of these in the body. Perhaps you've heard of the hepatic portal system before. This is one that's much smaller, but working on the same principle, and the principle is that we have blood flowing into a capillary bed. And normally, once blood goes from an artery into a capillary bed, it goes into a vein and back to the heart again to be reoxygenated in the lungs and then back out to the body. Well, in a portal system, what happens is once it leaves that first capillary bed, it's going to go through a portal vein that collects from that capillary bed and then delivered to another capillary bed again. And then once it leaves that capillary bed, then it has a vein that goes back to the heart and then can go through reoxygenation and repumping back out to the blood. So this is what a portal system is, and we have a portal system going on in the anterior pituitary. So what this means is that nearly all the blood that's going to the anterior pituitary has gone through the hypothalamus already in a capillary bed, so it's been in close contact with the hypothalamus, goes to the anterior pituitary, and then goes into another capillary bed where it's in very close contact with the anterior pituitary as well. And what this does is it allows the hypothalamus to provide hormones in high concentration, and very quickly to the anterior pituitary. So it can do this very, very quickly and get what it needs there in fairly high concentrations. So let's take a look at how this is happening. Again, we have the hypothalamus up here, which is part of the central nervous system. So again, we have our neurons here, which are going to then synapse in the infundibulum, or the beginning of the infundibulum here, onto this capillary bed. It's going to secrete some sort of hormones. Then they're going to be transported here through these pituitary portal veins to the anterior pituitary, where it goes through another bed of capillaries, where those hormones that were released by the hypothalamus are picked up, received by whatever target cells. That's going to affect some sort of reaction, and then the pituitary is going to adjust its release of hormones, either up or down based on that signal it gets, and then release those into the blood system as well. So that's how this connection works between the hypothalamus and the anterior pituitary. So most anterior pituitary hormones are tropic. So again, what tropic means is that these are hormones that are causing the release of other hormones, and there are 6 of them in total. So first off, we have growth hormone, or what's also known as somatotropin, and this is made by specialized cells called somatotropes. So what's after the colon here for all of these is going to be the specialized cell that is going to be secreting this particular type of hormone. And growth hormone is going to be, as the name suggests, regulating body growth. It has several other actions as well, but I'm only going to be relating to you the very specific major actions that each of these is responsible for. Now just kind of keep in mind that there's more actions that happen, but these are the main ones. We also have thyroxine-stimulating hormone, or TSH, and this is going to be made by thyrotropes in the anterior pituitary. And thyroxine-stimulating hormone is going to stimulate secretion of thyroid hormones, and it's also going to stimulate growth of the thyroid gland. We also have adrenocorticotropic hormone, or ACTH, and it's made by corticotropes. And this is going to stimulate cortisol release from the adrenal cortex, and it's also going to stimulate growth of the adrenal cortex. Kind of similar actions there. We also have follicle-stimulating hormone, which is a sex-related hormone, and this is going to be made by the gonadotropes. This is one of two different hormones that is made by the gonadotropes, one that we're going to talk about next. And in females, this causes the growth and development of the ovarian follicles, so important for reproduction there. Also in males, this is required for sperm production. So in both cases, it is one of the main hormones that is responsible for the formation of gametes. So then we have luteinizing hormone, or LH, also made by the gonadotropes. So this is the second one. The luteinizing hormone controls essentially sex hormone secretion in other parts of the body. Then we also have prolactin. That is created by lactotropes, and prolactin actually have very, very wide-ranging action. So the first action that we knew about is that it actually stimulates milk production, so that's why it gets the name prolactin. However, it also has other actions that relate to reproduction regulation, but also in osmolar regulation, growth regulation, water drive, other things like that. So this is a very wide-ranging, very important hormone that's being made there. So let's talk just a second. So we have these hormones that are being created by the hypothalamus, and hormones are being provided to the anterior pituitary by the hypothalamus, and control the secretion of the anterior pituitary. So we have these hormones that are being released by the hypothalamus, and these hormones are going to travel to the anterior pituitary and somehow control what the anterior pituitary is releasing. So for instance, we have thyrotropin releasing hormone. This is going to stimulate TSH and prolactin. We also have corticotropin releasing hormone that is going to stimulate the release of ACTH. We have gonadotropin releasing hormone that's going to stimulate follicle stimulating hormone and luteinizing hormone. We also have growth hormone releasing hormone that's going to stimulate the release in the anterior pituitary of growth hormone. We have growth hormone inhibiting hormone. So not all of these hormones that are going to be provided by the hypothalamus to the anterior pituitary are stimulating hormones. Some of them are actually inhibiting. So for instance, here, this is one of them. We're going to have this hormone inhibiting the release of growth hormone. We also have prolactin releasing hormone, which is going to stimulate the release of prolactin. So again, these are named for their primary functions, but one thing I want you to remember is that there's actually multiple functions for each of these. I'll come back to that. And then we also have prolactin inhibiting hormone, which is going to inhibit the release of prolactin. So we have this list of quite a few hormones that the hypothalamus is going to be releasing into the capillaries through the pituitary portal system to the anterior pituitary, and this is going to modulate the type of hormones that the anterior pituitary is releasing. And so this together, this chain of hormones is what's known as an endocrine axis. And this endocrine axis is this 3-hormone hierarchical chain of command. So for instance, the hypothalamus will be releasing thyrotropin releasing hormone. That's going to travel to the anterior pituitary, and that's going to cause the release of thyroid stimulating hormone, which will travel to the thyroid and stimulate the release of thyroid hormone. So really, we have this one hormone stimulating the release of another hormone, but stimulates the release of a third hormone. Why in the world do we do this? So it actually turns out, the thing you have to keep in mind, we're telling you the major actions. I'm telling you the major actions of each of these hormones, but this is not the only actions. Most of these hormones have more than one minor effect. And so what this happens is, like in the nervous system, we get divergence of signal happening in these hormones. We actually get one hormone that's going to stimulate majorly one other hormone, but also a few others as well. And then we can have divergence at each of these. But also, most pituitary hormones are actually controlled by more than one hypophysiotropic, so one coming from the hypothalamus, multiple hypophysiotropic hormones. And this allows for, not divergence of signal, but convergence of signal. So this allows for convergence of signal when we have multiple types of hormones that control, to some minor extent at least, the control of each of these pituitary hormones. So we both have divergence and convergence of signal because of these endocrine axes that we have, these three hormone hierarchical kind of chains that happen to cause hormonal regulation in the body. So let's talk a little bit about, so the hypothalamus is beginning this whole chain. How is that being controlled? Well, first off, the hypothalamus is part of the central nervous system. So it's getting all these neuronal connections from other parts of the brain that are helping to modulate its function, and the neurons of the hypothalamus can integrate those signals and elicit either action potentials or no action potentials that will go on to release neurotransmitter or neurohormone, whichever the case may be. So one of the main connections that's going on here is that the hypothalamus has close connections with the amygdala. The amygdala is connected with emotional processing, so you actually get some hormonal release that is connected with emotional processing, such as an animal becomes stressed, it's more likely to upregulate the cortisol axis, and we actually get release of cortisol, a stress hormone. But also, in some parts of the hypothalamus, there's no blood-brain barrier either, and this allows the hypothalamus to detect blood chemistry. Now, this is important because then the hypothalamus can adjust the release of hormone to bring blood chemistry back towards a set point and keep that relatively stable. So that's another way that some amount of control is happening with the hypothalamus. Also, we can have inhibition by target cells down the road. So the final hormone in some of these axes can inhibit the production of the first or second hormones, and thus down-regulating its own production if it gets to be too high. One of these examples is CRH will create ACTH or simulate the production of ACTH, which then in the adrenal cortex causes the release of the secretion of cortisol. So cortisol actually in this axis will inhibit the production of CRH and ACTH. So then if that gets too high, it's actually causing or preventing its own secretion. So that's really all I have for the pituitary. Let's move on to a couple of the other endocrine-only glands that we want to talk about. Now we're going to move on and talk a little bit about the thyroid gland. So the thyroid gland is located here on the anterior side of the windpipe, and it has these two lobes, and separated by this small little piece called the isthmus, between this little connection between these two lobes, kind of located below the larynx here. So that's where this next gland that we're going to talk about is located. And the thyroid is this major regulator of metabolism. That's really the most prominent function that it has. It has a lot of other smaller functions as well, but this is the major thing. And it's doing that through the action of several different hormones, two different hormones primarily. The first one is this one. This is tetraiodothyronine, or also known as thyroxine or T4. The big thing that, the reason this is called tetraiodo is that we have these 4 iodines here that are attached onto this. Now, one thing I want you to notice is that this is very similar to the structure of tyrosine. So over here, this is tyrosine, and it's actually made out of 2 different tyrosines is how this is going to be created. So if you look at just this part of the molecule right here, with the exception of those iodines that have been tacked on, this is exactly tyrosine right here. Essentially what we did is then pulled on to another tyrosine functional group here. So both thyroid hormones are essentially these modified tyrosine molecules that have had these iodine molecules or these iodine atoms knocked onto them. So the other one that we're going to talk about is triiodothyronine, or T3, again because it has 3 iodines instead of 4. Otherwise, the structure of T3 is identical to the structure of T4, just with 1 fewer iodine. So these are the 2 major hormones that the thyroid gland is going to be secreting, and together oftentimes they're just called thyroid hormone. So let's talk a little bit about how T4 and T3 are going to be stored in the cell. So the first thing that you need to think about is that both T3 and T4 are lipophilic, which means they're going to very easily diffuse out through cell membranes. They're going to be kind of hard to store. And for that reason, these molecules are going to be bound to a protein called thyroglobulin that will hold it in place and kind of sequester it inside the storage units of the thyroid gland so that it won't escape. And those storage units are actually these particular things called follicles. So a follicle is made up of these follicular cells arranged around fluid-filled spheres. Now, these fluid-filled spheres, try to say that 10 times fast, are filled with this liquid that is called colloid, which is mostly thyroglobulin. And of course, attached to this thyroglobulin is T4 and T3, those 2 hormones. And one interesting thing to note in this case about what's happening here is that this is an extracellular storage of a hormone. It's not being actually stored in the follicular cells, it's being stored in these spheres that the follicular cells are surrounding. So this is kind of an interesting storage mechanism for a hormone, unlike a lot of other ones. So let's talk about how in the world are T4 and T3 produced. And really, all of this synthesis is going to take place while bound to thyroglobulin, again, so we can hold on tightly to this lipophilic hormone. So the thyroglobulin is transported into the colloid after it's been produced. So we're into this extracellular storage space. And in that thyroglobulin, we have these tyrosine residues on there that we're going to be using to create thyroid hormone on that protein. So the tyrosine residues are on the thyroglobulin before it's turned into the T3 or T4. At the same time this is happening, the thyroid is capturing iodine from the blood and actually symporting it into the cell with sodium. So there are sodium, potassium, ATPases running, building up this concentration, this high concentration of sodium outside the cells, the follicular cells. And what we're doing is then symporting sodium down its gradient, but we're using that to power transporting iodine against its concentration gradient, because there's actually a lot of it inside these cells. So we're transporting it, symporting with sodium to get both sodium and iodine into these cells. And this is actually where essentially all of the iodine in the body is located, in the thyroid gland. And really, the creation of these 2 hormones is the only known function of iodine in vertebrates. This is really the only place you're using it. But that being said, even though this is the only place, these are really important hormones. So this is an important function of iodine. Iodine is really important because you need it for these hormones. Okay, so iodine is oxidized to produce the active form of iodine. So this is going to be happening through an enzyme called thyroperoxidase. And once this has been oxidized, we can now attach it to tyrosine. At times there will only be 1 iodine attached to tyrosine, in which case we get monoiodotyrosine, or MIT. Or there will be 2 iodines attached to tyrosine, in which case we get diiodotyrosine, or DIT. From there, we have a coupling of these iodinated tyrosine molecules to form thyroid hormone. So if we combine an MIT with a DIT, we get T3. So 1 with 2 gives us 3. Or if we combine 2 DITs, we get T4. Kind of makes sense. Well, let's talk about what happens to both T4 and T3 once they're produced and we want to secrete them out into the blood supply. Now remember, they are being stored extracellularly in these follicles. So we need to somehow first get them into the cell so something can happen. And how that happens is that these follicular cells are going to phagocytose a bit of this colloid. They essentially bite a chunk out of this colloid here to get some of that T4 and T3 into the cells. From there, we got colloid vesicles that from this phagocytosed colloid, and they're going to combine with lysosomes. And these lysosomes contain enzymes that are going to separate the T4 and the T3 from the thyroglobulin, are going to cleave it off of that protein. And once that happens, T4 and T3, being lipophilic, are easily going to diffuse out of the follicular cells into the bloodstream. This is going to be highly innervated with capillaries, so them just diffusing out of the follicular cells, most likely bring them into the blood supply of those capillaries running through that. So once they're into the capillaries, we have to get them somewhere. It turns out that they're going to be transported through the blood like most lipophilic hormones are, bound to some plasma protein. In this case, that plasma protein is thyroxine binding globulin. And almost all of the T4 and T3 are going to be bound to these globulins. So less than 1% of T3 and less than 0.1% of T4 are actually free in the blood. So very, very little of both of these are free in the blood. Most of it, the vast majority of it, is bound to these plasma globulins. T4 will also bind to albumin. So albumin is one of these kind of just general transport plasma proteins that will just kind of hoover up whatever lipophilic proteins are out there. And T4 will also bind to this albumin, but T3 won't. Okay, so once it's out in the bloodstream, what is going to happen to T4 and T3? Well, it turns out most of the T4 actually is converted to T3 in peripheral tissues. So most of that is being taken up, and you're chopping off an iodine off of this T4 to make T3. And this is happening by a series of enzymes called the deiodinase enzymes. And it actually turns out that T3 is about 10 times more active than T4 is. So it's much more active. And this deiodination of T4 turns out to be one of the major regulatory pathways of thyroid hormone. So remember back when we were talking about how we regulate the amount of hormone that's active in the blood at any one time. One of those regulatory steps or regulatory processes, strategies, was how quickly you convert an inactive form to an active form. This isn't quite an inactive to an active form, it's a less active to an active form, but this is one kind of shining example where we have that sort of activation being a major regulatory step in the endocrine system. So, now that we have the active forms, or the very active forms, T3 ends up, and T4, regulate basal metabolic rate. This is the primary way that basal metabolic rate is set in animals. Now, we'll come back and we'll talk a lot more about basal metabolic rate later. This is really just kind of like how much metabolic activity is happening at your resting state. But the way that T3 and T4 does this, the primary way that it does this, is by regulating the activity of sodium, potassium, ATPases. Now that might seem something small, but it actually turns out that somewhere between 20 and 40% of the energy usage of a cell, which in turn means 20 to 40%, a big chunk of your whole metabolism, is being dedicated to running these sodium, potassium, ATPases. This one pump is one of the major uses of energy in your body. It actually turns out this is producing most of your metabolic heat at rest. So this is where most of your metabolic heat is coming from. This is what's being regulated, the primary thing that's being regulated by T3 and T4, which means that T3 and T4 are regulating to a large degree how much heat are you creating. T3 and T4 are also going to somewhat regulate metabolic fuels. So if it's regulating the metabolic rate, it makes sense that it also plays some role in regulating how much metabolic fuel you have supplied to that metabolism that you're trying to fire. So most of this is happening by the synthesis and breakdown of fats, proteins, and carbohydrates. It actually turns out that both of these molecules, both of these hormones can actually fuel both synthesis and breakdown of a lot of these, depending on the concentration that it occurs in the plasma. It also has a permissive action with catecholamine, so it's going to cause the proliferation of catecholamine receptors in target cells of catecholamines, making them more sensitive to the catecholamines, like norepinephrine and epinephrine. And what this ends up doing is it's called a sympathomimetic activity. When you give T3 and T4, it can appear as if it's having the same action as the sympathetic nervous system would have. It's not doing that exactly. What it's doing is it's making peripheral tissues more sensitive to the sympathetic nervous system. But it looks like, or mimics, the actions of the sympathetic nervous system. So that's really going to wrap up what we're talking about with the thyroid gland. It's really just a very quick overview, and we're going to come back to both this when we talk about metabolic rate and metabolism and energy processing, because it's really important in that. But for now, we're just going to leave it at that and move on to the adrenal gland. Okay, let's go ahead and finish up the endocrine system by talking about the adrenal glands. And these are located right above on the superior side of your kidneys. That's actually what they mean. Ad means above, and renal means and renal means kidney. So these are the above kidney glands. So just really briefly talking about their anatomy, the first part that is important is we have the adrenal cortex. This is the outer portion of the adrenal gland. It's producing steroids, and it's producing these steroids in these steroidogenic cells. And really, the adrenal cortex is making up approximately 80% of the gland in most mammals. We also have this other portion called the adrenal medulla, which is the center portion of the adrenal gland, and it's producing catecholamines. So again, catecholamines are the neurohormones such as norepinephrine, epinephrine, and actually dopamine is lumped in there as well. And it's going to make these in some specialized cells called the chromaffin cells that we'll talk a little bit more about in a little while. Okay, so let's talk about the different kinds of things that are happening in the adrenal cortex specifically. Then we'll move on to the adrenal medulla after that. So the adrenal cortex is making several different classes of hormones, like the mineralocorticoids, which includes aldosterone, also the glucocorticoids that we'll talk a bit about. And this includes hormones like cortisol and corticosterone. Then we also have sex steroids that are going to be made in the adrenal cortex. So this is your things like androgens and estrogens, your what are typically called the masculinizing and feminizing sex steroids. And then we also have this other major one that has a fairly big effect, particularly in males, which is the androsterone. Anyways, just shorten out to DHEA, and this is a fairly important one. I'm not going to really talk about the sex steroids right now because I'm planning on talking about that a little bit more later when we talk about reproduction. But for the moment, we'll just kind of leave it at that. All of these steroids, again, remember, they're lipophilic, and they're all going to be circulating bound to plasma proteins. And they vary to how much of those are going to be free and how many are bound. Now, none of these are nearly as readily bound to their plasma proteins or in such high levels bound to their plasma proteins as the thyroid hormones were, but it's still pretty high. So for aldosterone, it's somewhere in the neighborhood of 30% is free. For the glucocorticoids, somewhere around 10% is going to be free in the plasma. So let's start out talking a little bit about the glucocorticoids and what's going on with them. So they have a good number of metabolic actions. So for instance, one thing that they'll do is they'll stimulate gluconeogenesis in the liver. Gluconeogenesis, again, is the creation of glucose from non-glucose molecules. This is one way to mobilize energy from non-carbohydrates. It also inhibits glucose uptake by most tissues, except for the brain. It keeps glucose uptake happening by the brain, and this is a phenomenon known as glucose sparing. So as you start to get low on energetic or metabolic fuel molecules like glucose, you need to save most of your glucose for your brain, because your brain is really, really reliant on glucose, more so than most of your other tissues. And what that means then is that in this glucose sparing, we're going to stop glucose use by most of your other tissues, and they can move on to something else. They can start using fatty acids and other things like that. And we're going to save those glucose molecules for the brain to use. That's glucose sparing. It's also going to stimulate protein degradation, particularly in muscle cells. And it's also going to stimulate lipolysis, so the breakdown of fatty acids. Altogether, all of these metabolic actions that we were just talking about are all working to mobilize energy stores. All of these are taking your stored energy and making sure that you have them mobilized and ready to use. So really, this is kind of working together to fuel metabolism, and fuel it in a way as if you were starving or something like that. There's also some permissive actions that are happening by the glucocorticoids, including they permit catecholamines to induce vasoconstriction. So constricting arterioles, and this will generally cause blood pressure to increase. There's also a very strong activity of glucocorticoids in modulating the long-term stress response. And so a stress response is just, as your book defines it, this is generalized non-specific response to factors that might overwhelm your body's ability to maintain homeostasis. So, for instance, a lot of different things can cause this. You're starving, something's trying to eat you, you're falling off a cliff. I mean, a lot of different things. Anything that's going to tend to kill you or injure you or make you not have a good day. Those are the kinds of things that are going to threaten to overwhelm your body's ability to maintain homeostasis, and that could be bad. And these things that do this, these things that induce the stress response, are known as stressors. They can be physical, so for instance, your arm got lopped off. It could be chemical, you have no oxygen, or something else like that. It could be physiological. When your arm got lopped off, you're losing a lot of blood. You have low blood pressure. That's not helping. It can be psychological. Something could simply be causing fright, or something else like this, like loud noise, continued loud noises, something like that. Or it could also be social, and social animals, if they're put into situations that are degrading their social status, or in such a way that separates them from their social group, this can also induce stress as well. And so all these stressors are going to result in increased glucocorticoid secretion. And it's going to, again, it's not exactly certain the one-to-one relationship between these, but it's going to tend to mobilize your metabolic fuels so that you can even continue to fuel your body. And so it's somewhat intuitive that you might need these in stressful situations, in a fight-flight sort of situation, or if you're starving, something else like this, you might need your internal body stores of energy to be mobilized. But there's a lot of times where it's not really all that adaptive. For instance, if it's simply an ongoing long-term social stressor that's happening, mobilizing your energy source might not necessarily be the best adaptive response. So it's not exactly beneficial to always increase this sort of stress response to all the different stressors that actually do cause this. Also, glucocorticoids play a role in inflammation suppression. So it actually turns out that glucocorticoids, particularly cortisol, keeps inflammation response in check during injury. So that's good. If you have some sort of injury, it's good to not let the inflammation response overwhelm other processes going on and get out of check. And cortisol is going to interfere with most of the steps in inflammation. So this is actually acting in a lot of different places. However, this has some interesting kind of side effects. What this means is, together with the stress response, that we're mobilizing these during stress responses, animals under constant stress tend to be more prone to infection, because these same hormones that are mobilizing metabolic fuels are also depressing inflammation responses and interfering with the immune system, therefore making these organisms that are under stress more prone to infection. Okay, so that's the adrenal cortex. Let's move on a little bit to the adrenal medulla here. And really, it's pretty interesting. The adrenal medulla really is just a really big sympathetic ganglia. It's just a modified big sympathetic part of the extension of the sympathetic nervous system. So if you remember us talking, it has these specialized chromatin cells for producing the catecholamines that's going to be secreting into the blood supply. And really, chromatin cells are really just modified post-ganglion sympathetic neurons. So it's being innervated by these presynaptic or pre-ganglionic sympathetic neurons that are synapsing onto these chromatin cells that are really just modified post-ganglion sympathetic neurons. These chromatin cells then are able to release norepinephrine and epinephrine directly into the blood supply, basically doing the same thing that post-ganglionic sympathetic nervous or nerve cell would do, releasing norepinephrine onto the target cell. However, that being said, epinephrine is a very major component here, is what's predominantly secreted here. And this is not actually a very common neurotransmitter that's used in the post-ganglionic sympathetic nervous system neurons. So this is incredibly increased in these chromatin cells and somewhat unique to them. Stored in and all these neurohormones, these catecholamines, are going to be stored in these chromatin granules until they're exocytosed. So really, these chromatin granules are just modified, essentially, neurotransmitter vesicles. They're going to be exocytosed into the bloodstream, and that's how we get the secretion here. Now, one thing to keep in mind, this secretion is controlled completely by the sympathetic nervous system. There's not really anything else that's playing into how this is secreted. This is simply the sympathetic nervous system, which makes sense. If this really is just an extension of a sympathetic ganglion that's gone wild, essentially, grown up to massive proportions, it would make sense that the only thing controlling it then is these inputs from the sympathetic nervous system. Okay, so let's talk about what happens during catecholamine release. So during maximal sympathetic stimulation, you get this massive dumping of these catecholamines by the adrenal medulla into the plasma, and it can actually increase the plasma catecholamine concentration 300-fold. That's a lot. And what these are going to do is these are going to very quickly circulate throughout the circulatory system and coordinate a sympathetic response. So help coordinate the sympathetic response to whatever is stimulating. So we get norepinephrine in one of the components there, and this is going to bind to alpha and beta-1 adrenergic receptors. You probably remember these from when we're talking about the sympathetic nervous system. And is going to bind to these. Now, epinephrine also binds to these as well. Now, norepinephrine is going to bind with higher affinity to the alpha cells, the alpha adrenergic receptors, whereas epinephrine and norepinephrine bind basically equally as well to the beta-1 adrenergic receptors. Now, that being said, there's one more type here, these beta-2 adrenergic receptors, and it turns out that these are only stimulated by epinephrine, not by norepinephrine. And so this is actually good because we can get a varied response by some cells based on what kind of adrenergic receptors they have and what catecholamines we've released into the blood. So, for instance, activation of most alpha and beta 1 receptors, especially alpha 1 receptors, in arterioles is going to cause vasoconstriction in those arterioles. Vasoconstriction is going to cause an increase in blood pressure, which is what you want during essentially sympathetic nervous system input. But you don't necessarily want that everywhere. You don't want vasoconstriction happening everywhere. And you don't want to have that happening in skeletal muscle and in the heart. You want to increase blood flow to those areas. So the stimulation of beta 2 receptors by epinephrine in skeletal muscle and heart muscle arterioles is going to cause vasodilation. The actual opposite response as what we had in the alpha-1 adrenergic receptors that would respond to both epinephrine and norepinephrine. So this allows us to have differential responses of different tissues, but to essentially the same molecules. Okay, so really we're going to just kind of finish up there with the endocrine system, and after the test, we're going to come back and start talking more about metabolic processes, energy processing, stuff like that. So, let's finish up there.